So today's talk, I'm just going to focus on carnivorous plants, which, as I said, I have a particular love for. Shown here is a Venus flytrap, and you can see in this picture I took, we have this insect prey here, this fly, which has been captured by the leaf of this plant and is being slowly digested over time, and the nutrients absorbed to acquire nutrients which the plant is otherwise not getting from the nutrient-poor soils in which it grows. So I think that these are really remarkable, interesting plants. However, right, I'm a staff scientist in the lab of Chory at Salk, and her lab is not interested in carnivorous plants per se, but rather they're interested in this, and this is how plants interact with their environment. So a plant, right, unlike us as animals, a plant is sessile, so wherever that seed lands and germinates, that's where the plant has to grow. And it needs to sense then everything within that local environment, whether it be light signals, for example, shade from other plants, day length. It needs to sense water availability, nutrients, diseases, herbivores, and so on, and then it needs to mount a response to those. And for plants, that response is generally a developmental one. So you're probably all familiar if you take many species of plants, right, and grow them at home, and you put that pot near a windowsill, sometimes you'll see that plant starting to grow towards the light. So that's an example of a developmental response to an environmental signal. And one environmental signal which plants sense is mechanical stimuli. And mechanical stimuli can take different forms. For example, you can imagine a root system, right? So Todd showed these images of roots, and these roots need to grow down through the soil, through this soil substrate, and there they'll be touching components of the substrate, and they can respond to this. Another mechanical signal perceived by plants, which we often don't think about, is salt. Salt will cause changes in cell swelling due to osmosis and the water of movement, and the movement of water in cells. So this is another form of a mechanical stimulus. And then also, imagine, for example, if you go out in, I think, La Jolla, Ellen Browning Scripps Park, if you've been there and you see these trees which are being swept by the wind, so constantly touching a plant, for example, by wind, will cause these developmental changes as well. And you start to see that the trunks get thicker, the plant's a little bit shorter, and this is a process known as thigmomorphogenesis. So plants do perceive mechanical stimuli. The question then, right, what our lab is interested in is how they do this. How do they perceive mechanical force? And we're a molecular genetics lab, so we're particularly interested in the molecules which are involved in this particular process. And to address this question, again, due to my interests, I like to turn to extreme model plant systems. And here I turn to carnivorous plants. And the reason for this is that carnivorous plants often have some very rapid responses to animal touch because they need to catch a fast-moving animal prey to make a meal of it. So shown here, does anyone know this particular species of plant? Someone does?
Sundew.
Yeah, so this is a type of sundew. I don't know if you're familiar with it. For those who will rotate through the lab with me, I'll show you some sundews there. So this sundew has on it, this is the leaf of the sundew. It's a pretty bizarre looking leaf. Has anyone been to the San Diego Zoo? They've got a carnivorous, we have a plant lover back there. At the carnivorous, at the San Diego Zoo, they have a little carnivorous plant greenhouse, and you can see examples of pitcher plants, Venus flytraps, and lots of different species of sundew there as well. So this is the leaf of the plant. And then coming off the leaf, you can see these little hairs, which we commonly call tentacles. And these tentacles secrete at the tip of their hair this little drop of a bioadhesive glue, right? And so an insect comes and lands on the leaf, and then it gets stuck to this little drop of glue. And then it starts thrashing around, and the plant recognizes the touch signals from the insect, and those tentacles start to bend over the insect, drowning it in glue, and then ultimately all those touch signals and also some chemical signals, they cause the plant leaf to curl over the insect, and then you get this open stomach forming there. Those tentacles will secrete a bunch of digestive enzymes, break down the soft parts of the insect and absorb nutrients. So this is a real life plant monster, right? And what you can see there this very dramatic behavioral response almost, right, to a touch stimulus. When we look at the molecular level, we can also see some quite rapid responses to touch. So here now what you're looking at these tentacles here, and this is a special plant. So this is a transgenic sundew plant now which I've created, and basically I've put what we call a reporter into this plant, and again we'll talk about this for those who come to my rotation. We put a reporter in this plant which is going to fluoresce when calcium is present. So calcium ions are very common intracellular signaling molecules in plants and animals, and they often signal the state of the environment within the cell. And so this reporter is going to start fluorescing in a green color when calcium is abundant in a particular location within the cell. And so what you can see here, I'll play this video, when we should loop. Oops. So it's paused. Let's play it again. Right, so we're going to touch the tip of this tentacle up here, and you can see when we touch it, you see this wave of calcium going down the tentacle, right? So you see at the morphological level, you see these responses to touch, and then you can also see these responses, which are quite rapid at the molecular level. And those responses can be small and localized, as was shown there, or they can be very large. So here now we're doing a harsh touch stimulus to the leaf of one of these plants, and you now see this systemic wave of calcium going through the plant in response to that signal. So somehow the plant, right, it perceives these different environmental cues and mechanical signals, and then it needs to mount an appropriate response. So that's all I'm really going to talk about sundew for today, is just a little bit of a teaser there. If you're interested to learn a little bit more about that piece of work, Education Outreach here at Salk helped put together a video which we put out for World Carnivorous Plant Day last year, and you can learn a little bit. Yeah, it's a real thing. I know. These plants certainly they inspire the imagination, right? So I encourage you to look at that if you're interested. Okay, so what we're really interested in then, and the story which I thought I would talk to you about today is, right, what is the molecular basis? So you've seen this response to touch. What is the molecular basis for prey touch sensation, right? What are the molecules which are actually, and the proteins which are involved in sensing that initial touch stimulus? And for this, we turn to an even more, certainly I would think the most famous carnivorous plant. So this is the Venus flytrap. For those who are unfamiliar, I'll play the video, right? So here, this insect has been attracted to the leaf by color and volatile emissions, and once there, it starts wandering around on the leaf, and then it comes into contact with one or more hairs, right? And then bang. So it touches these hairs, they're called trigger hairs, and then that signals to the leaf to close over the prey. So this is no longer, again, it's no longer a slow developmental response to the environment as you would see in most other plants, but rather we have this very extreme, rapid, bistable response to touch. And due to the extreme nature of this response, I would argue that it now provides us this unique system for understanding how plants can sense mechanical signals from animals. All right, so here, this is a close-up of one of these hairs on the leaf, right? So you can see this hair here. So it has this long lever, and then it sits on this podium. An insect bumps into that hair, and then what happens as a result of that is you get this electrical wave propagating across the leaf. So this was really first proposed by Charles Darwin. So he did quite an intensive study on these plants, and one of his colleagues, who was an electrophysiologist, animal electrophysiologist at the time, was the first to record these electrical waves in the Venus flytrap. And we now know that this wave takes the form of do you guys know what this particular electrical signal is? It's an action potential, right? So this has basically the same shape as the action potentials and the impulses which go through our own nervous system. By contrast, though, it is a lot slower. So we have this electrical signal, right, which goes through the leaf. And in biology, electrical signals are associated with ions, right? And it's the movement of ions across membranes, which moves charge then across membranes. So this is where the electricity is coming. So this requires ion channels, right? So these are proteins which are embedded within the cell membrane, and they can move different salt ions such as potassium, sodium, calcium across the membrane to move charge, right? So we're going to focus now on ion channels. And what we wanted to find then was ion channels which might potentially be involved in sensing that initial mechanical stimulus. So here, this is a little bit more of a complicated experiment. So this is using what we call next generation sequencing technologies, basically ways to sequence DNA at a massive scale. So this is what Todd's group works on. So here what you're looking at, right, this is all the genes which are expressed within the trigger hair. And here on the y-axis, you're looking at the enrichment in expression of those genes in the trigger hair relative to the underlying leaf tissue. And then they just spread across the x-axis by some relative measure of gene expression. And what we find is that there's three particular genes which stood out to us as having very large expression, relative expression within the trigger hair relative to the leaf. And these are also illustrated over here. And they all code for ion channels. Two of them code for ion channels which we called flycatcher1 and flycatcher2, or flyc1 and flyc2. So flyc1 was very massively enriched within the trigger hair, and then the third is an ion channel from the Oscar family. Okay, where might these channels be expressed in the trigger hair? So here, this is an electron microscopy image of the base of the trigger hair. This is the lever coming down. Here's the podium. Here, between the lever and the podium, is kind of some ruffled cells. This is known as the indentation zone, and this is where the mechanosensory cells are present. So this is where you get maximum bending of the trigger hair when an insect bumps into it. And what we find is that at least one of these ion channels, flyc1, is expressed almost exclusively within those sensory cells. So this now is a method known as RNA in situ hybridization. Basically, what you want to look at here, right, this is a cross-section through the trigger hair. All these little red dots here, these correspond to RNA transcripts of that flyc1 ion channel. And you can see that basically these little red dots are localized within this indentation zone within the sensory cells. So these ion channels, right, they're expressed within the trigger hair. They're expressed within the right place to be involved with touch sensation. And the reason we're particularly interested in them is that we noticed that these flycatcher1 and flycatcher2 ion channels, they shared similarities with a very well-known mechanosensitive ion channel actually from bacteria called mass gas. So in bacteria, mass gas is required to protect the cells from osmotic downshock, right? If you took a bacterial cell and you dropped it into a low salt environment, water would start to move into the cell through this process of osmosis. The cell would swell and then it would pop. So this channel senses that cell swelling and it opens, allowing the free flow of ions across the cell membrane to equilibrate the salt concentrations and prevent the rupture of the cell. So what our results might now suggest then, right, is we've taken this ancestral osmosensing ion channel and we've put it to this new purpose, and that's sensing touch within the venous flytrap. So here, I just thought this was a cool video, so I would play it. This is not work of mine. This was work from my collaborator, Artem Pardapudian at Scripps. This is the structure of the flyc1 ion channel. That hole you saw was the pore going through the membrane. These alpha helices here are embedded within the membrane. These ones here, which are parallel to the membrane, they might help gate the channel open and close in response to membrane stretch. And what they find, right, when they take this ion channel and they put it in mammalian cells, which is what they've done here, so take that venous flytrap ion channel, express it in a mammalian cell, and then they apply membrane stretch to that mammalian cell. So this is the pressure stimulus here, which they apply through a recording electrode, and then they see when the membrane is stretched that you get these individual flyc1 channel opening and closing events, right? So this channel can form a functional membrane stretch activated ion channel. Is it actually involved in touch sensation? So to test this more formally, I come from a lab which is mostly based in genetics. It's the major tools which we use. So what I did was to go in and try to mutate these genes, the flycatcher1 and flycatcher2 genes within the Venus flytrap genome. So we're creating here some transgenic monster plants, I guess, if you want to call it that, right? So here we're using CRISPR-Cas9 technology. Have you guys heard of CRISPR-Cas9? So it gets a lot of press nowadays, won the Nobel Prize recently. There's a lot of talk that maybe it will come into play for gene therapies for humans. It's really remarkable technology, and we take advantage of this in our lab as well. For those who are unfamiliar, the idea is that you have this protein enzyme, which is called Cas9. So this is a DNA cutter. And this enzyme then, it gets targeted to a specific sequence of DNA through this other molecule which it associates with called a guide RNA. So this guide RNA has two components to it. It has this blue component which binds to Cas9, and then it has this green component which is complementary to the target gene which you want to mutate. So in my case, the FlyC1 and FlyC2 genes. And then when Cas9 gets targeted here, it will cut the DNA. The cell recognizes this cut and it tries to repair it. And in that process of doing the repair, sometimes it makes some errors. And you get small deletions and small insertions. So this is the method which we're using. And so I mutated both those two flycatcher1 and flycatcher2 ion channels. So as of to date, these are still the only targeted genetic modifications for any carnivorous plant. The plant, you can see here, looks morphologically normal. And then, surprisingly, what we found, and admittedly initially quite disappointing, was that these plants are, sometimes it happens in science, right? You do the right experiment, but you don't always get the result which you expect. These plants responded to touch quite normally. So here we're doing a harsh touch stimulus, basically 2 touches manually with a micropipette, and you can see that the leaves respond to that harsh touch stimulus quite normally. So at this point, again, just disappointed, we sat back and we scratched our heads a little bit, and what we thought was that maybe we were missing something here, that this stimulus which we were seeing here was too strong. So the trigger hair is exquisitely sensitive. It can sense an angular deflection of 3 degrees, right, less than 3 degrees. And here we've knocked out 2 ion channels. We know that there's at least one other ion channel, that Oscar ion channel, which I showed on a previous graph. So, right, you knock out 2 of them, maybe the other one is still functioning. So maybe, or there could be other ion channels we haven't yet identified. So this is very common in biology. We call this redundancy in the function of genes and proteins. And so maybe what we needed was a more sensitive assay to look for a defect in mechanoperception in these plants. And one of the wonderful things about Salk is that you can, there's no real departments here and structure in that regard, so you can go talk with other people around campus. My background is developmental neurobiology and genetics. So I had a previous friend and colleague here, Shrikant Chalasani, who runs a neurobiology lab here at Salk, and they are interested in what they call sonogenetics. And I'll discuss that a little bit later too. So the assay which we turned to in collaboration with Shrikant's group is an ultrasound-based assay. And the idea here is that you can take these detached Venus flytrap leaves now, right, and you can put them on an ultrasound transducer. This will, you can then apply an ultrasound, right, this is a mechanical wave, right, high frequency sound wave. You can apply that to the leaf and you can keep ramping up the intensity of that wave until the leaf closes. And so I'll just play that video here. Right, and so you can see eventually it reaches a point where the leaf closes. And what we now find, so this now gives us a very quantitative sensitive assay, and what we now find using this more sensitized assay is that we have this defect in these double mutant plants in response to this mechanical stimulus, right? So this is consistent now with these ion channels functioning redundantly with other mechanosensitive ion channels in mechanoperception within the trigger hairs. And so the model which we proposed, right, is that bending of this trigger hair at this region here, where fly C1, fly C2, Oscar, and perhaps as yet other unidentified ion channels are expressed. It causes membrane stretch there. This opens the ion channels. You get the movement of ions then across the membrane. This depolarizes the cell and then sets up this action potential, which then propagates through the leaf, right, through these little passages, conduits, which connect leaf cells together. These are called the plasmodesmata, right? So these, and also voltage-gated ion channels, right? So this electrical signal propagates through the leaf and then causes the two leaf lobes to close by an as yet unknown mechanism. And what I think this story reinforces again is this idea of co-option in evolution, right? We're taking this ancestral ion channel and we're putting it to this new purpose. So this ion channel was originally involved in sensing osmolarity in bacteria, and now plants, at least carnivorous plants, have put it towards touch perception. And it remains to be seen then the roles of these ion channels in other plants. So there are groups which studies this particular family of mechanosensitive ion channels in plants. They seem to have some roles in pollen rehydration, right? So when the pollen lands on the carpel and it gets rehydrated, it starts to swell. These ion channels are involved in sensing that swelling and making sure that the pollen cell doesn't die as a result. There's Oscar channels involved in salt sensation. Whether or not these fly C channels are involved in some of these other processes remains to be seen. And then there's the last slide, which I think is rather a fun way to end and a little bit sci-fi. It's a little bit out there. But I like this because it illustrates that I'm a basic biologist. I love basic biology. I love just learning about nature and the world around us. But sometimes what we discover can have unintended applied benefits as well. So what Shrikant's group is now doing is taking some of these ion channels which we're discovering in plants, and he's putting them into mammalian cells. And these ion channels, as I showed, these can be regulated by ultrasound activity. So you can imagine now that you can regulate the activity of these mammalian cells by ultrasound. So these mammalian cells, they might be insulin-secreting pancreatic cells. An example which Shrikant really likes is, let's say, a neuron which senses pain, which is inappropriately firing. And now you can put this ion channel in there. You can apply some ultrasound, and then it will act to alter and silence the activity of that cell and dull that pain sensation. So there might be some applied medical application here, and this is something which is being looked at. So again, I like this example because it shows where basic biology can sometimes lead to. You just have to have an open mind. So to end, a big shout out to my boss, Joanne. She's been very encouraging. As I said, I like some weird, cool plants, and by and large, she's allowed me to follow my interests. These are some of the key collaborators. I've already pointed out Shrek. Again, similar to others, this actually is a high school student, Charlotte, who worked with me, and she actually helped generate those beautiful calcium reporter sundew transgenic plants, which I showed at the beginning of the talk. So a big thank you to her. And thank you, and I'd be happy to take any questions. Yeah.
Is there a difference in photosynthesis rates between carnivorous plants and non-carnivorous plants?
I don't know if there's a difference in rates. I'm sure someone's looked at that. By and large, photosynthesis for these plants is not limiting. So most carnivorous plants, not all of them, but by and large, most of them grow in very open bogs. And so there's plenty of sunlight, plenty of water, right? So photosynthesis can just run amok. What is limiting is nitrogen and minerals. These are all being leached away by the water. So carnivory, because it is costly, right? There's a cost to making these leaf structures and going through this process of digestion. By and large, photosynthesis is unlimited to them, right? If if you want to think of it that way. The cost, right, but they are limited in nutrients, and so therefore they're willing to invest to get the nutrients they wouldn't otherwise get. Yeah.
Sounds like some great science fiction stuff.
Yeah, it does, doesn't it?
So I have a question about the relationship between the original ion channels in bacteria and how they got into plants.
Mhm.
How did that?
I mean, this might be a better question for Todd. I mean, genomes, right, are so they're not found in animals, I should say that, right? So this particular family of ion channels, it's found in bacteria, it's found in plants, although it's only that pore region which is conserved. Otherwise, if I was to show you the bacterial channel and the plant channel, you might not immediately see the relationship, but just the pore is pretty well conserved. You know, whether it was adopted at some other point in time, right, that the DNA moved into a plant, although I think it's pretty ancient. I think they're all the way down. So I assume it's when plants first evolved, this gene was carried in them. So I don't know if people have looked at some of the more ancestral plants to see how far back it goes, but I think it's present in all of them. Yeah.
Can you talk a little bit more about your high school curriculum that you developed?
Yeah, so the idea here is that teachers were interested in this Harnessing Plants initiative, which Todd talked about, obviously, and students are interested in it too. They're interested in climate change. It all sounds very catastrophic and doomsday in a way it is. But at the same time, I think one of the nice things about the Salk program is that can we think of interesting and novel ways to tackle this very large problem. And so the idea was for students to learn a little bit of the science which we're doing, but also to do some hands-on experiments and learn how we grow plants, how do we measure things like root traits, how do we see how plants respond to the environment, and so to bring that hands-on material into the classroom for students to work with, to do hopefully an experiment, and in that regard to also fit with these, what are they, the next, you guys would know better than me, the next science standards. Yeah. What they call next generation, next generation stuff nowadays. Next generation science standards, right? So that they can, hopefully, this was the intention, that they can be a part of directing those experiments. Yeah.
When you mentioned about inserting ion channels into mammalian cells, are they considering that more for nerve blocks or possibly for long-term nerve relief, chronic?
Yeah, so I mean, we're now getting into the realm of gene therapy, and I love the idea of this. There are some roadblocks in that this would require taking a plant channel and putting it into an animal, and the FDA usually frowns upon moving things from non-human organisms into humans. So the idea is that you would be expressing this channel, right? And due to the properties of this channel and why Shrikant's group is particularly interested in it, in plants it activates cells, and in animals, due to the ions which it conducts, it will actually function to silence cells. So in that regard, it's a little bit unusual. All the other mechanosensitive channels which Shrikant's group studies, which he's collected from animal systems, they all, in response to ultrasound, they will all activate the cell, and this one will silence. So it gives him a novel, what he calls a sono-silencing tool. So the idea now is, yes, if you have if it's something like chronic pain, and then it's due to a neuron which is, a pain-sensing neuron which is inappropriately firing, right? You can imagine you could have an ankle bracelet or something, you could press a button, you could apply the ultrasound as you need to, and then it would shut down that neuron and prevent it from being active to silence that sensation of pain. So again, it sounds sci-fi, but it's cool. It's a cool idea to think about, but there are certain roadblocks to actually implementing it. It might, though, make, even if it doesn't find its way into humans, it might be an interesting set of tools which can still be applied for people doing mouse research or just interested in neurobiology, for example, where you can start to do experiments where you target specific parts of the circuit, knock it down, and see what effect you have on the animal. Yeah.
I'll ask the last question and keep it really silly. If you had to be a carnivorous plant, which one would you be?
Which one You know, I'd like to be more creative, but the reality is the Venus flytrap is the coolest of them. It's not the fastest actually. There is a faster carnivorous plant which grows in water. It's called a bladderwort. That one functions a little bit differently because it's more like a mouse trap, right? It's like a spring loaded. It's held under vacuum. The animal hits it, and then it opens it, and the vacuum just sucks the animal in. So that's the fastest known moving plant there is. But there's just something iconic about the Venus. It looks like a monster, right? I mean, so it's a remark. I mean, you look around at nature, and nature does some pretty remarkable stuff sometimes. So yeah, I'd have to go to the Venus flytrap.
Thank you so much.